So welcome back friends, welcome back in another video tutorial from microbiology with Shaknik. So in that video tutorial we are going to discuss about the regulation of tryptophan operon, that is the positive regulation and negative regulation. Okay, so in our previous video I have introduced all the genes that are important in tryptophan operon, right? All the regulatory genes and the structural genes, and I have also told you in that video that there is a gap present in between regulatory operon and structural operon, and as a result their configuration become 3 o'clock configuration, right? So today's our installment is all about regulation of the tryptophan operon. Okay, how they become positively regulated and negatively regulated, and in that video I am going to compare that topic with lactose operon. Okay, so let's start our discussion. So now for some time let's go back to the lactose operon and find out what are the default choice of lactose operon. So lactose operon's default choice is negative regulation. Okay, that means when there will be a RNA polymerase in the promoter of the regulatory site, it will facilitate the transcription of the repressor gene, and it will promote the production of protein. Okay, but those protein are initially a inactive protein, as you can see, and these inactive proteins are called aporepressor. By nature they are inactive. Clear? So they will initially cannot bind. That is the case in case of tryptophan operon. Sorry for that, we are talking about lactose operon. So for now let's consider this operon as a lactose operon. So in lactose operon the RNA polymerase will bind with the promoter of the regulatory part and it will start transcription. Okay, so there will be too much amount of lac-I production, and lac-I will facilitate the production of a repressor, which is a active repressor. Okay, now those active repressor will go immediately toward in the operator and prevent RNA polymerase from transcription. That is the by default choice of lactose operon that we have discussed, right? That is the negative regulation also. But when there is some amount of lactose present in the cell, this lactose will be converted into allolactose with the help of trans-acetylase present synthesized by lac-A gene. Okay, clear? Now those trans-acetylase will convert lactose into allolactose, that is the beta-1,4 glycosidic linkage will simply converted into beta-1,6 glycosidic linkage here. Okay, now those allolactose will bind with the active repressor of lactose and prevent and convert them inactive, right? And as a result they cannot bind with the operator and RNA polymerase can start its transcription. That is the positive regulation, right? So here as you can see that lactose operon is induced by allolactose to start its transcription, right? So what can we say? We can say that allolactose is an example of inducible operon. Okay, similarly in case of tryptophan operon, the mechanism is just the alteration of the lactose operon. Remember, in case of tryptophan operon, the by default mechanism is the positive regulation. Okay, so first of all RNA polymerase will bind with the promoter, that was I actually telling at the starting of that video. So the promoter will first bind with the RNA polymerase will first bind with the promoter of the regulatory site and it will start transcription, and after transcription it will gives to the production of inactive aporepressor. Okay, that is the meaning of aporepressor. You know that aporepressor is nothing but inactive repressor. There are repressor that are by nature inactive. So as a result, they will not unable to bind to the operator, as you can see. So operator is free initially, so RNA polymerase can happily start its transcription, produce genes and synthesize tryptophan, right? That is the positive regulation, and that is their by default choice. Okay, now in what is the negative regulation, let's discuss. In case of negative regulation, remember, as their by default choice is positive regulation, they will start transcribing and translating, so that too much amount of tryptophan is synthesized, right? So those tryptophan when there is a too much amount of tryptophan present in the cell sorry, this tryptophan will go and bind with those inactive aporepressor, okay, and convert them into active repressor, as you can see. And those active repressor have a high binding affinity towards their operator site of the structural part, okay. So they will bind over here, and as a result, RNA polymerase now experiencing a obstacle in its way, so it cannot transcribe happily, right? So active repressor TRP bound prevent their transcription, okay. So remember here the most important thing, that in negative regulation, TRP activates those aporepressor which are by nature inactive and make them active repressor, okay. That is the concept behind the negative regulation, okay. Now you tell me what kind of operon they are, okay. Although I have already written here, but just try to think about this, that in case of tryptophan operon, their initial choice is to start the operon, right? The default choice is to start the operon, but when there is a too much amount of tryptophan, this excess tryptophan can repress their transcription, right? So can we say that this operon is an example of repressible operon, isn't it? So that is the idea. So remember one thing, I am wrapping up their video. So in case of lactose operon, their default choice is negative regulation, and in case of tryptophan operon, their default choice is positive regulation. Lactose operon is induced by allolactose, right? So it is an example of IO, that means inducible operon. And in case of tryptophan operon is repressed by too much amount of tryptophan. So can we say that it is an example of repressible operon? Obviously. So that is the concept behind the positive regulation and negative regulation of tryptophan operon. So MCQ may came that what is the default choice of tryptophan operon? The answer will be positive regulation, because here the repressor is inactive, right? So it cannot bind with the operator initially, so RNA polymerase can transcribe happily, right? But whenever too much amount of repressor is present in the too much amount of tryptophan is present in the environment, this excess amount of tryptophan have the ability to convert those inactive repressor into active repressor, and as a result they can happily bind with the operator and prevent RNA polymerase from transcription, right? So tryptophan operon can be repressed by excess amount of tryptophan, so their default choice is here positive regulation, and they can be repressed, that is by repressible operon. And today's video just fulfill our first day's question, that lactose operon will be on when lactose is present in the environment, right? Because conversion of allolactose is necessary to inactivate the repressor to start transcription, right? And on the other hand, tryptophan operon will on when tryptophan is not present in the environment, right? Because if tryptophan is present in excess amount, it will immediately activate those aporepressor into repressor, so that tryptophan will not start their transcription, okay? So that is the concept for today. So let's summarize what we have learned. So LAC TRP. So LAC is a catabolic operon. There is a conversion of lactose into glucose and galactose. Tryptophan is an anabolic operon, okay. In case of tryptophan operon, you have leader sequence that will facilitate transcription attenuation. We are going to discuss this in our upcoming video. Lactose operon do not have any kind of leader sequence, right? In case of tryptophan operon, is a repressible operon. Why? Because tryptophan, excess amount of tryptophan simply repressing the tryptophan operon, right? And on the other hand, lactose operon is inducible operon. The presence of lactose will convert themselves into allolactose, and allolactose will inactivate the repressor, and as a result lactose operon can start in transcription. Okay. And by default choice of lactose operon is negative, and by default choice of tryptophan operon is positive regulation. Okay, so that is for today, friends. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. We will again come back with another topic. So till then, bye bye.